Bismillahirrahmanirrahim. In this paper, we are going to just discuss, in this video, we are just going to discuss a few paper 4 skills just before the exams, and I hope this helps you, this video helps you. Recite Surah Fatiha 3 times, pray to Allah for guidance in every exam of life. This is a very small exam, just a bio paper 4 or something like that. But in life, you will come across many hardships in life, many decisions to take, many decisions to decide your life journey. Always remember, ask Allah for guidance, and Allah will never ever disappoint you. I just want to help you in really figuring out how to answer the question 1, which is usually in a certain pattern. This, of course, is from 0610 paper. Now, I'm just going to read this to you, and then I'm going to tell you how you're going to answer this question. Rice contains starch. Amylase is an enzyme that catalyzes the breakdown of starch to form reducing sugars. A student investigated the effect of amylase on cooked rice. The student used this method. Step 1. 2 small beakers were prepared. Each beaker contained 20 grams of cooked white rice. One beaker was labeled W and one beaker was labeled A. Okay, so draw this. One beaker W, one beaker A, and you put in 20 grams of rice in both of them. Both of these were kept in a water bath. Water bath means it is a big tub and you have water in it, and it would have a thermometer in it, and it would be measuring the temperature, and we kept the temperature at 40 degrees Celsius in the water bath. So the rice in both was at 40 degrees Celsius. A measuring cylinder was used to add 20 centimeter of distilled water to the beaker in A. So here we added distilled water to the one in W. So we added how much? 20 centimeter cube of distilled water into this in beaker W. The same measuring cylinder we use then again. So first we put water in the measuring cylinder, and then we put was used to add 20 centimeter cube of 1% amylase solution. Remember, amylase is an enzyme. First we added the water and then we used the same measuring cylinder. This again is static in the question. If you had done the other way around, what would have been wrong? Think of it. You would added amylase first, and then some amylase was still sticking to it, and then you added distilled water. You didn't wash it, but you added distilled water first, and then you didn't wash it, and then you added amylase to it, and then you added this to beaker A. This was beaker A. The beakers were left in the water bath for 10 minutes. Now imagine you're doing this in the lab. I don't know if you've ever been to the lab, but I take my students to the lab. One test tube was then, so you've left this for 10 minutes. Now you take two test tubes, another two test tube. One test tube is labeled W10 and one test tube is labeled A10. You label it with a glass marking pencil, which is given to you, a glass marking marker. So one test tube is labeled W10 and one test tube is labeled A10. After 10 minutes, this 10 minutes, a glass rod was used to stir the contents of beaker W. So with a glass rod, we stir this, mix it up like you stir tea. Stir the contents of beaker W. A clean pipette was used to remove 2 centimeter of the liquid. So we removed 2 centimeter out of this, 2 centimeter of the liquid surrounding the rice in beaker W, and we put the 2 centimeter here. From W, from here, we put it here now. The 2 centimeter of liquid was put in W10. Now what did we do? Step 7 and 8. This step 7 and 8 then we repeated with beaker A. So again we took 2 centimeter from here and we put it here in A10. So we've taken the liquid. It says the liquid surrounding the rice in beaker W, and then the liquid surrounding beaker A, and then we put it in another test tube. Now we used a syringe and we added 2 centimeter of Benedict's. Now Benedict's, you know, is a sort of a blue color. So I don't have a blue color difference, so I just use this color. So then we put 2 centimeter of Benedict's in both of them. Each of the test tubes W10 and A10. Test tubes W10 and A10 then were put in a water bath. Then we put this in a water bath at 80 degrees Celsius. and we put this for 5 minutes. Now read everything very carefully, do this sort of a drawing which I am doing on the paper here, and then we left it for 5 minutes. After 5 minutes, the color of the contents of W10 were observed. Observed. Please remember, what does it mean to observe and what does it mean to conclude? Observe, you are late to the class. Conclusion, you didn't get up in time, so you are late, which happens nearly every day. Some of you are so good at that. The students' observation are shown in figure 1.1. W10 started blue and stayed blue. A10 started blue, became orange. Right? Now, please remember that you should do this just like as I have told you. Now I will do this again in this diagram. I made that on top of that, but now I do it separately. Two small beakers were prepared. Each beaker contained 20 grams of cooked rice. One beaker was labeled W and one beaker was labeled A. So you can see this here and here. Both of the beakers are put into a 40 degrees Celsius water bath. A measuring cylinder was used to add 20 centimeter of distilled water. So we added this to this. The same measuring cylinder was then used to add 20 centimeter of 1% amylase to this. The beaker would left in the water bath for 10 minutes. We left it for 10 minutes. One test tube was labeled, then we have another one. Then we say after 10 minutes, one test tube was labeled W10 and second test tube was labeled A10. After 10 minutes, a glass rod was used to stir the contents of beaker W. So we used a glass rod to stir this. A clean pipette was used to remove 2 centimeter of the liquid rice in beaker W, and the 2 centimeter was then put here in W10. 2 centimeter cube of the liquid. And steps 7 and 8 were repeated with beaker A, and so we added 2 centimeter from that here as well. So if you do this sort of drawing, then you sort of know that this is the practical, because please remember, when you're doing a paper 4, somewhere in some other country people are doing paper 3s and they're doing this experiment in the lab. So they're going to be examined for that. So you've got to have a similar sort of an exam. And then we kept this in a water bath at 80 degrees Celsius and we left it for 5 minutes. And then after 5 minutes we see the color. In W10 it remained blue. In A10 it became orange. And what did we add? We added Benedict's solution. You know, Benedict's is for reducing sugars. So this means there was no reducing sugar here, but there was reducing sugar in this one. That is, of course, the conclusion. Observation is remain blue, change color to orange. Then it says prepare a table to record the result shown in figure 1.1. So usually you see the main thing is that your heading should be separate from the heading should be separate. And the mark scheme also says that 2 columns, rows with lines separating headings from the body of the table. Here there were no units, but usually the units must be written here. Like for instance, if you had time, then you would write either minutes or seconds. Whatever is given in the question, then you would give those units. So this would go in the heading. Of course, in this question, there's no time in minutes, but I'm just telling you, I'm just giving you an example of it. So prepare a table. Then it says, state a conclusion for the result shown in figure 1.1. So what is the conclusion? You could have said rice contains starch and that is broken down to reducing sugar. Or you could say starch is broken down by amylase and a reducing sugar is produced. So conclusion means that the one which contained amylase has resulted in the formation of a reducing sugar. Well, why? Because rice contains starch. And when you added amylase, amylase breaks down the starch to maltose. and maltose is a reducing sugar. Starch to maltose, and maltose is a reducing sugar. So state the conclusion. Conclusion means what did you conclude? If you don't know the English, then you must look up what the word conclusion means. Observation means what do you see? Conclusion means what is your result? What have you learned from it? Like I talked to a student today and I found out what was my conclusion. My conclusion was sorry, he's a high level chungar. I know you all laugh at that and say, minus you call everybody chungars. So yes, my conclusion was that he's a high level chungar. Part 3, it says state two variables that were kept constant in this investigation. Now you see, variables that were kept constant in the investigation are usually in the question. And you can see in the question that there were many things which were kept constant. What was it? The mass of rice was kept constant. Where is that? 20 grams of rice. Mass of rice was kept constant. What else was kept constant? Type of rice. Cooked white rice. So the type of rice was kept constant. So the mass of rice was kept constant. The type of rice was kept constant. It said white rice here. Then temperature of the water bath was kept constant, 40 degrees Celsius. Then time in the water bath that we said 10 minutes. This was kept constant. Everything's in the question. Volume of water and amylase solution was kept constant here, 2 centimeter of the liquid. Here, this of course when we take it out, but 20 centimeter of amylase and 20 centimeter of distilled water. So volume of water and the amylase solution. Then the sample size removed, 2 centimeter of the liquid removed. We didn't just say take out a few drops, or we just say with a syringe take out a little bit of the water surrounding the rice. So 2 centimeter. And then we said very clearly, we said 2 centimeter of the Benedict solution. So all these things were given in the question. All these things were given in the question. You've got to pick these up from the question. What were the variables that were kept constant in this investigation? Please be very careful in writing these, because these are not 2 marks that you should lose at all, because it's all given in the question. I've only written 2 of the points, but all the other points I've explained to you earlier. Now, part 4, state the purpose of adding distilled water to the beaker of cooked rice labeled W. Why did we have one with amylase and one with water? The water one is for a control. And why do we have a control? Control is for a comparison, because this is, when biologists disagree. I always had to explain this to a student, and he just did not understand what a control meant. So I said, okay, when 2 biologists are arguing with each other, I didn't say fight. I said when they are arguing with each other and they say, okay, well, it's not the amylase, then the other one says, okay, let's do without the amylase and just add water to it. And if it still works, then that means amylase was actually not the thing which was causing all the conversion of starch to reducing sugar. So control is for the comparison to show that what would happen if no amylase were present, or to prove that the color change was due to the amylase. So please understand what is a control. Control is for when 2 biologists are disagreeing on something. And we say, okay, well, somebody says, okay, well, amylase is going to break down the starch to maltose. And I say, no, well, it's not going to break it down. I think even if you add water, it will break it down. So when we 2 biologists are disagreeing, then we say, okay, let's do the experiment with the amylase. Let's do an experiment without the amylase. So as a control for comparison, right? Okay, then we come to part 5. It was important that step 3 was carried out before step 4. Predict the effect on the result if step 4 was carried out before step 3. What was step 3 and step 4? Go back and read it. A measuring cylinder was used to add 20 centimeter of distilled water to the rice in beaker W. The same measuring cylinder was used to add 20 centimeter of 1% amylase to the rice in beaker A. And when I was reading this, I told you about this. Now, what does it say? It says predict the result if step 4 was carried out before. You see, then we say W10 will change color to yellow or orange because a positive result in the test tube. Why? Because you see, if you've taken first you used the amylase and then you used You had a bit of the amylase still sticking to it. So the one with the W10, which was the one that only had water in it, that would also change color because some amylase will go in the W10 as well. So some amylase would go in that. So there would be some reducing sugar in the W10 as well. Because if you did that first, then that means some amylase would still remain and it would go into W, in which you are supposed to only add distilled water. But with the distilled water would be a little bit of the amylase as well. So W10 would also have a color change to orange. Then coming to part 6, the temperature at the 40 degrees Celsius water bath was not maintained and decreased during the investigation. State one piece of equipment that could be used to maintain the temperature at 40 degrees Celsius. Well, that's very simple. Thermostatically controlled water bath. You use a thermostatically controlled water bath. I said this to you to remember this as a very important terminology, which is electrically controlled water baths actually. Thermostatically controlled water bath. Or what you could have said, thermo-isolated container, insulated jacket or insulation, or you say lid used on the water bath. So anything which maintain the temperature, of course the electrically ones are of course very, very good and they maintain the temperature. Then it says, state how you could show that cooked rice contains starch. How would you say, what do you do? How do you prove something has starch? What do you do if I say, okay, potatoes contain starch? So you take a bit of the potato, crush it up, add iodine solution, and if it turns blue black, well, it's got starch in it. I say to check a piece of apple for starch. Do apples have starch in it? Well, you just crush a piece of the apple and add iodine solution, and it will become blue black, and that means starch is present. So add iodine solution, blue black color if starch is present. Then as we come to these sort of questions where there's a graph, some students investigated the effect of temperature on the rate of amylase activity. They measured the mass of reducing sugar produced in 5 minutes at different temperatures. The results are shown. Now what do we have? We have mass of reducing sugar produced in 5 minutes in milligrams, and here we have temperature. So you see this at 10 degrees. How much is the mass of reducing sugar in milligrams? Nearly about 12. Then it keeps on increasing, and then let's look at this one. It's about 45. What is the temperature here? Let's look at it. It's 35. So always look at the graph, always see the peak or see the beginning, then see there's an increase and then there's a decrease. There could be an increase and then there could be a plateau. So you see how it is. Maybe, it's something like this. These are different shapes of graphs which you all must understand and comprehend. Now it says describe the effect of temperature on the activity of amylase. Please remember, describe is what? It's no biology. You do not give me any biological explanation. It's no biology. So you just look at the graph. Let's go back to the graph. And it says describe. Describe means no biology. Explain means biology. If you have to say explain, then you have to give me biology. So what is happening here? A very simple thing. The activity of amylase or the mass of reducing sugar is increasing and then decreasing. So it's increasing and then decreasing. Basically, it went up and then it's decreasing. So the activity of amylase, or you could say the mass of reducing sugar, increased and then decreased as the temperature increased. And what is the peak? Peak would be the optimum, and that temperature is 35 degrees Celsius. So this is a describe. Describe means everybody should get their full marks in the describe. In A levels, describe is very profoundly a lot of marks, worth of a lot of marks. And if anybody gets all those correct, you're sure to get a better grade. So describe is no bio, it's just an explanation of the graph. Increase, then decrease, or in this case, you would have said increase, then plateaus. And then you would have given me this point where it starts to plateau. In this situation, you've given me increase and then decrease, and then you give me the peak at which which is the optimum temperature, which is 35 degrees Celsius. Either you said mass of reducing sugar, or you said the activity of amylase increased and then decreased, and the peak and the optimum temperature. So this is 1 mark and this is the second mark. Use a graph to estimate the rate of amylase in milligram per minute at a temperature of 42 degrees Celsius. And space for working, there are 2 marks for this. So you would have to do this on the graph. So you'd say at 42, and then you'd sort of draw a line and something like this, and you would check this out. And it was 42. You see, this was in 5 minutes. So 30 over 5 is 6. And this is where the student would have got this wrong. I'm sure many students would have got this wrong because they didn't read the 5 minutes. So 30 milligrams you would have said, and then it would have said rate per minute. You didn't read this properly. So it is 6 milligram per minute. Milligram per minute was given in the question. Then it says the student wanted to obtain a more accurate value for the temperature at which amylase works best. Suggest further investigation work that the student should carry out. Now, you see, this is something which you have to understand. You can't do this question if you haven't understood this. It said obtain a more accurate value. What does the word accurate mean? You see, many of you read the mark scheme, and the mark scheme says test at smaller intervals of temperature. What does that mean? Some of you don't even understand that. Some of you, of course, do understand. I'm not saying that, but some of you don't understand and you don't bother to, go into detail and ask yourself. So you see, they took a figure at 30, and then they took something at 35, and then they took one at 40. Now, what if they had taken at 32 and they got a point here, and then they took at 34 and they got a point here, and then they took at 35, and then they took at 36 and it was somewhere here, and then at 38 it was somewhere here. So you see how the graph would change. The graph would become like this. So what does it mean when it says how would you get a better estimate of the accuracy of it would improve? Test at smaller intervals of temperature. Here we were traveling every 5, 30, then 35, then 40. Then we would do at 30, 32, maybe 34, 36, 38, and then 40. Now, here now we only had 30, 35, and 40. Then we would have these. So this is called test at smaller intervals of temperature. Please understand the English. Some of us are very pathetic in English. Or you set a test at smaller intervals of temperature, and you could select a narrower range of temperatures around 35. Around 35, because we've got 35 we know is the optimum. So around 35. Some less than 35 and some more than 35. So please remember these wordings. These wordings are very important. Test at smaller intervals of temperature. We can have a question on pH, and you can say test at smaller intervals of pH, and select a narrow range of temperatures around 35. If we got in the pH, it was something like around 7, so then we would say around pH 7. So test at smaller intervals of temperature and select a narrower range of temperatures around 35 degrees Celsius. This completes this video on which I have explained to you how to properly handle question number 1, which is going to come in your paper 4, whether you're doing 5090 or 0610. These are some steps which I want you to know for both exams, which is on Tuesday and coming very soon. So best of luck, and please give it your best. Thank you so much.